But again, it's what the drug companies want. They
Right.
don't want exercise because it's going to take away market share. They don't want good diet because it takes away market share. So they love that, message to go out. But I think it's completely misleading.
Welcome to Longevity Roadmap. This is Buck Joffrey. And, today's a really interesting show. It's about mitochondria. From everything I've read and heard, I suspect that, frankly, that the major advancements in longevity medicine over the next few years may come actually in the form of drugs or perhaps even procedures that improve mitochondrial health. I'm personally, I've kind of been focused on this myself because I do think it is a key part of healthspan, lifespan. Taking a few supplements. I've been taking CoQ10. I've been taking N-acetylcysteine. That's NAC for those of you who are interested, which you'll actually hear about today on today's show. But do I feel any better taking these things? No. But, the reality is I don't really necessarily expect to, because I don't think that, taking these supplements in general, I don't think that, realistically that you are going to have some sort of miraculous change in the way you feel. Some people do. I acknowledge that, but I wonder how much of that is really placebo, because most of this stuff is going to be subtle stuff happening at the cellular level. And for me, a lot of the supplements are really in hopes of some sort of level of prevention, Honestly, apart from, hormone replacement therapy, I don't really think there's anything that is going to, that you're going to take necessarily is going to take an immediate change in quality of life or, muscle mass or anything like that. But HRT is a big one though. So if you are, if you are in your 40s and 50s and you're feeling sluggish and not well, or if you're a woman, you're, postmenopausal and you're just not yourself. I mean, there's plenty of, there's so much out there right now just to get you back to normal. So I encourage that. But let's get back to mitochondria. When you think about restoring mitochondrial health, what are you doing, right? Mitochondria, as you might remember from high school biology books tell us, from what the biology books tell us, are the powerhouses of the cells, right? And if we could get them back to the way they were functioning, maybe in our 20s, maybe we'd feel different. We'd feel better, more energetic, something like that. And I'm hopeful that we can get to that reality sometime here in the future, because I do think that interventions, successful interventions focused on mitochondria probably would make an enormous difference. Now, today's show, again, is with an expert on mitochondrial health. You hear people in the longevity space talking about mitochondria all the time, and I'm convinced that a lot of them don't even really know what mitochondria are, because they tend to, talk about it like, I don't know, that like sort of drives me crazy, frankly. The likes of Dave Asprey. I mean, listen, there is a lot of that we need to learn about this space. We do know a lot already. And the point I'm making here is that I want to try again to keep this show focused on real science. And I want to make sure that you get a chance to learn from an expert in this space. And the one that we've got today, he's got an MD, sees patients, but he's also a PhD as well. So we get the clinical and scientific parts covered, all in all. And we will have that interview when we come back from these messages.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen. And it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to the show, everyone. Today, my guest on Longevity Roadmap is Dr. Mark Tarnopolsky. He's a clinician scientist specializing in neuromuscular and mitochondrial medicine. a professor of pediatrics and medicine at McMaster University and director of the Neuromuscular and Neurometabolic Clinic at McMaster University Medical Center. He has an MD and a PhD in cell biology and metabolism. He's published over 550 peer-reviewed papers, and he's the founder and CEO of Exokine Corporation, which is a biotech company developing innovative therapies for aging, obesity, muscular dystrophy, and mitochondrial dysfunction. Mark, how are you doing today?
Well, thank you. And yourself?
Good. I'm doing well. Thank you. I'm excited to talk to you. This is an area where, I feel like, when it comes to longevity science, longevity medicine, it seems like there's each few months, there's like a new area that seems to like get attention. And at least in my view, mitochondria have been sort of really hot lately. Obviously they're really hot for you all the time because that's what you're knee deep in. But let's talk a little bit. This show has a lot of science people and physicians, but it also has just smart people interested in science. But why don't we start with this? The mitochondria are, they're known as the cell's powerhouses. They're basic organelles sitting outside of the cell's nucleus. How do mitochondrial structure and function change with age?
Yeah, so mitochondria, as I'm sure familiar with, are present in every cell in our body with the exception of mature red blood cells, and they're particularly enriched in tissues that require a lot of energy. So we have them in hearts, we have them in muscle, we have them in brain, in fairly high abundance. In fact, almost 20% of heart tissue is actually made up of mitochondria because it's constantly working. So as we get older, mitochondria have a very sort of intimate relationship from a structural functional perspective in our tissues, and they come together. That's a process called fusion. They break apart. That's a process called fission. And when those processes are impaired, as they are in some of our patients, we see massive alterations in the structure of mitochondria, which then is very well correlated to the functional consequences. So with aging, we do have a reduction in the total mitochondrial mass. Again, it takes a while before we really start to see that decline, but we start to see alterations in their ability to come together and to break apart. What happens is when we have dysfunctional mitochondria, we sort of wall off the areas that are more damaged through a process called mitophagy, which is a special form of what's called autophagy or self-eating, where we remove the damaged parts. And over time, much like pruning a tree in the springtime, if you can't cleave off those bad areas, the mitochondria become abnormal in terms of their function related to the structural alterations. And so that impairment of autophagy or slash mitophagy is part of what we see with aging, which then leads to decrease in the function. And in addition to supplying energy, they have many other consequences for the cell. They're involved in apoptosis, our telomeres, they're involved in inflammation, oxidative stress. So many of those buzzwords that people associate with aging can ultimately be related to mitochondria as the proximate cause of so many of the other factors that are associated with human aging.
So one of the paradoxes of mitochondria is that we need them, and when they function really well, they're providing us energy, but they're also constantly throwing off what are called reactive oxygen species, which ultimately are things that end up also damaging mitochondrial structure as well. Can you talk a little bit about sort of that paradox of, and how that manifests over time as a person ages?
Yeah, so very much like the concept of inflammaging or the chronic low-grade inflammation that happens with aging, we also can have low-grade elevations of oxidative stress. So what happens, for example, when we exercise and we stress the cell, especially when there is no oxygen, we call that anaerobic metabolism at the beginning, then we bring in oxygen and we really stress the muscle, especially in the recovery if we really push ourselves and get some damage. We actually can get a production of free radicals from the mitochondria, especially at complex 1 and 3, which are two of the main engines of the mitochondria. Those free radicals can in excess do two things. One, excessive amounts can cause actual damage, and the mitochondrial DNA is very close to that source of generation, and it's particularly sensitive because the mitochondrial DNA doesn't have an area called introns where you can kind of buffer some of the damage, and the repair mechanisms are a little bit suboptimal. So when we get too much oxidative stress, it can in turn damage the mitochondrial DNA, which then in turn can affect the function of some of the complexes which are transcribed from that mitochondrial DNA and perpetuate this, what we call vicious cycle. And that concept was put forward many, many, many decades ago by a fellow named Denham Harmon. There's quite a bit of evidence we won't get into now that supports that concept.
Yeah.
However, a really important point that I want to get across is that the pulses that we get of oxidative stress coming from the mitochondria, for example, with exercise, are actually good. So that's part of the pruning of a tree hypothesis that I call it. And that is that those pulses of oxidative stress can cause some damage, and the areas over a period of time which start to accumulate damage in the mitochondria are then identified as being, for example, suboptimal. And that's one of the signals to prune the tree, get rid of the dysfunctional mitochondria. So at the end of multiple stresses, you're left with better mitochondria. And that concept comes into sharp focus when we think about the ubiquitous application of antioxidants. So there was a study years ago where they used vitamin E and vitamin C to suppress these free radicals, thinking, this is a good thing.
Yeah.
And these were in, people who were pre-diabetic. The problem is that sometimes those signals can function as a adaptive signaling pathway. So what happens with those pulses of free radicals with exercise, we stimulate the biogenesis of the mitochondria through something called PGC1 alpha. And at least in the one study that was published by Risto, R-I-S-T-O-W, was the first author, they actually showed that they could blunt the adaptive benefits of exercise, giving certain antioxidants. So, it's a double-edged sword. On the one hand,
yeah, interesting.
On the other hand, chronic low grade is not good.
So I guess the question is, like, when it comes to that, like, how do you balance that? Do you pulse the antioxidants and, so that they don't overlap with, exercise induced beneficial reactive oxidation species generation? Or how do you do that in theory? How ideally?
Yes, I think there's two answers to that. Number one, if we have, for example, disease like the people I see with muscular dystrophy or genetic mitochondrial disease, their levels are chronically very elevated. And so provision of antioxidants can lower the abnormally high reactive oxygen species and bring it to a more physiologic range. So that then you can allow for this pulsatility and adaptive benefits, which we know even in our severe mitochondrial patients, they can adapt to exercise. So on the one hand, and it may be the case with advanced aging that, we get to a point where there's this higher chronic low grade oxidative stress, and suppression of that to a physiologic range is a good thing. It's very much like what we call a Starling curve. If you suppress on the way up, it's not a good thing. But if you're into that downward spiral, suppression brings you back to a higher level. And so that concept is a tough one for people to really conceptualize and to think of when it may be beneficial. So for an athlete, for example, I know I've pushed myself as I used to be an elite athlete, many points into damage, into pathology, where I couldn't walk downstairs for 5 or 6 days. That caused so much damage. Adventure racing, where we would be out for 3 or 4 days. Clearly, excessive ROS and inflammation was generated. And I think during those situations, antioxidants are beneficial. The other interesting concept, too, is not all antioxidants are created equal. And so until I have seen a specific mix of antioxidants given to a human being and see what the effects are in that human being, both at rest and in the context of exercise, then I'm more confident saying that this is something I prescribe to my patients, or as I do personally take myself.
So is it fair to say that right now, maybe the better option is try to strive towards things that induce mitophagy and healthy mitochondria, rather than necessarily thinking about blunting, because there's a lot of use of antioxidants, right? Like, that's what I'm trying to make sure people kind of get an understanding of it. Like, is there a point to them taking these antioxidants at the end
Yeah,
of the
it's
day?
a difficult one. Number one, I think there's absolutely no ambiguity or question that if you have deficiencies, replacing a deficiency makes sense.
Yeah.
And so, for example, it's rare, but we do see vitamin E deficiency in about 4% of our patients. Vitamin B is a little different. It's not so much an antioxidant, but that's a very common vitamin to be deficient. Vitamin D has ubiquitous properties. And so we see a lot of those deficiencies. So replacing deficiencies makes sense. Deficiencies of antioxidants like vitamin C are becoming more common, interestingly enough. We
Huh.
certainly see it in
Yeah.
kids. Yeah. Kids with autism and such, where they become very restrictive in their eating. And I suspect that other people with very restrictive eating patterns,
Like the keto
they're
diets and
they're not
stuff.
measuring it, but they're
Yeah.
actually probably
Yeah.
getting deficiencies. So in those cases, measure and replace deficiencies. Next, um, y- there are so-called antioxidants that people can take, which in the human body can actually function as transient pro-oxidants. And certainly, we've
Mm.
spent a lot of time looking at this because we were very worried that giving antioxidants to people would blunt the benefits of exercise, which is really, the only proven way to extend health span and lifespan is regular exercise. Say what you may about a nematode or a mouse or whatever. In humans, the
Yeah.
only proven lifespan extension is regular exercise. So we don't want to ever blunt that benefit. So in our hands, we have combined what are considered antioxidants with exercise and actually showed enhancement in some capacity. And it may be that transiently they're working at a pro-oxidant capacity and then an antioxidant capacity. So it's much more nuanced and complicated, I think, than people really want to think. But whatever someone chooses to take, make sure that it's been studied in a human in a clinical trial and it does not blunt the benefits of exercise.
It might be useful, I think, to use the example of some of the rare mitochondrial disorders like MELAS, MELAS,
Mhm.
to kind of give a sense to people like what are the symptoms? Like what happens when things go really badly with mitochondria? And I guess maybe you could create some sort of parallel with what happens over time in aging or with some age-related diseases.
Yeah, the mitochondrial disorders are a little bit unique because you can have, mutations that are very close and they can have very different clinical consequences. But some of the things I think that resonate best with aging is high frequency sensory neural hearing loss. So that is what all of us experience. And there's these really wonderful apps that you can go where there's a high pitched sound. And as it drops down, it pretty much plus or minus 2 years can tell you what your age is. I
No
remember.
kidding. Yeah.
Oh yeah, my
Okay. I didn't.
daughter, when she was a teenager, we were going to ski races, and they put this thing on. The kids would all be freaking out because they could hear it. My wife and I couldn't hear a darn thing, and those really are very, very sensitive markers of your biological age, plus or minus a couple of years from. So what happens is the higher frequencies, those high pitched sounds, is what we start to lose as we age. And our patients, they're getting that sometimes even as children. So that is clearly, the optic nerve, the nerve that transmits our hearing, is very sensitive, and we see that in aging, and
Yeah.
that's such a common correlation. Next, things like cataracts. So pretty much everyone's going to have some degree of yellowing of the eyes and eventually cataracts when they're over the age of 65. Again, our patients, they're getting cataracts much earlier. So when I see cataracts in a 20 or 30-year-old, I'm thinking, this could be a mitochondrial disease. Those are two really classic features. What we
Yeah.
also see is I've got some pictures of some of my patients with their permission, of course, and the graying of hair, the wrinkling of skin, unbelievably advanced in many of our patients. And the big thing that really resonates, I think, is the exercise intolerance, lower VO2, and muscle atrophy. So every human being, I used to be an international level endurance athlete. My VO2 has dropped by about 30%, and all of us will be going down over time. Of course, if you start higher as an endurance athlete, you're going to, be at a better spot when you're 60 or 70, but VO2 will go down. And that's your ability of the heart to pump and your muscles to extract oxygen, which is largely proportionate to your mitochondria. So those go down. And we see that in our patients. They have profoundly low VO2 max. And then, of course, muscle atrophy. people have clearly shown that with aging in rats and humans, that the muscle fibers that start to accumulate mitochondrial dysfunction become atrophic and smaller. And that's one of several contributors to the sarcopenia aging process. And that's also occurring in nerves as well. So those, I think, are probably the most common things we see in our patients, and all of us will experience as we age.
So there are some organs, for example, like the brain, where you have this, you don't really have mitosis. You don't have a lot of cells replicating over time once you get a certain age. Same thing with the muscle. So do you see a difference in terms of mitochondrial effects on those organs where they're potentially accumulating mitochondrial damage over time compared to, like, say, I don't know, skin cells or whatever, that are turning over constantly and presumably have a new source of mitochondria?
Yeah, so it's interesting that you mentioned muscle and skin. So with muscle, we and others have clearly shown that you can do endurance exercise and resistance exercise and clearly show benefits, mainly improved VO2 max and mitochondria with the endurance and increased strength with the resistance exercise. But even with weight training in older adults, early on, one of my graduate students, Johnny Parisi, clearly showed that we improve the antioxidant capacity of muscle. We increase the mitochondria. And what's interesting is even though you're pulsing the body and increasing oxidative stress, eventually we lower oxidative stress in the muscle. When we first started these, people said, oh, you should never exercise train older adults. You'll actually cause damage. But in fact, we actually lower the oxidative stress. So it's clearly a benefit in those older adults to exercise muscle. Now, skin, interestingly enough, we did some studies published back in 2015, I recall, where we did punch biopsies of young people, older adults. We did this in a non-sun exposed area to remove that confounding effect. So it was in the upper buttocks where it is not really sun exposed. And we showed, first of all, that the chronic exercisers, so-called masters athletes, had much better mitochondrial function in their skin. But importantly, it's never too late to start. So clearly, after 3 months of endurance training, we showed improvements in mitochondrial function and even the thinning of the dermis. So that's your layer that has all the collagen. That's why people, get wrinkles like me and they stick collagen in there to plump things up with more collagen. That clearly goes down with aging, but we showed that even after 3 months of training, we could thicken that collagen. So there's definitely both effects on the muscle that's exercising, but, we release these factors, various chemicals, exosomes containing proteins and such, which then have a systemic benefit. And we clearly showed that in the skin.
So you can make a difference, obviously, then in muscle, and presumably that's cardiac muscle as well, heart muscle as well. And is that correct?
Yeah, that's absolutely true. In animals, they've shown that as well. And we know cardiovascular disease, which is more blood vessel disease, is lower. But even things like cardiomyopathy, can improve, which is something that people with hypertension can get, a thickening of the heart. So some of those factors definitely can be better. Yeah.
What about neurodegenerative disease, like Alzheimer's disease? What is the role of the mitochondria? What do we see with mitochondrial changes and patterns there? Because again, we're talking about an organ where those. I don't know how much mitochondria you can change there, but why don't you talk a little bit about that?
Yeah, so there's a variety of what we call neurodegenerative disorders. Some are genetic, some we don't know if probably isn't a monogenetic cause for them. Alzheimer's being one that's not usually a monogenic disorder, although there are some mutations which cause it.
Mm-hmm.
Parkinson's disease, there are some toxins, there are some genetic mutations and things like Parkin, which is involved in this mitophagy process, which can lead to these disorders. So Huntington's, Parkinson's, Alzheimer's disease. When people look in animal models and when they're fortunate enough to get brain samples from humans, there's clear evidence of mitochondrial dysfunction. Some can be a primary mitochondrial disorder. So, for example, some of the mitochondrial DNA mutations can give a very Parkinson-like clinical manifestation. But whether it's idiopathic Parkinson disease or Parkinson secondary to mitochondrial disease, at the cell level, there's only so many ways a nerve cell can die. And that's apoptosis, oxidative stress, mitochondrial dysfunction, inflammation. And so targeting those final common pathways, all of which are favorably enhanced with exercise, even in the brain, we can get neurogenesis and some benefits as well.
So you've got a mitochondrial cocktail that you've developed, and understanding it's a sort of mixture of CoQ10, creatine, and alpha lipoic acid. Can you talk a little bit about like sort of what each one of those does? And I'm assuming there's some sort of synergistic activity there.
Yeah, so in 2001, Flint Beal, who's a neurologist who does a lot of work more with the things I discussed, Huntington's and Parkinson's. I do more metabolic genetics, but we got together and published a paper where we put forward the hypothesis that to treat complex neurologic disorders and aging, people trying to simplify things, that there's really final common pathways that we need to target. And those pathways are apoptosis, oxidative stress, mitochondrial dysfunction, inflammation, and which all can contribute to decreased protein synthesis, blah, blah, blah. So using a single agent, like an antioxidant, vitamin E, for example, you're not going to target all of those pathways. So we hypothesized that single agents would be inferior to a multi-ingredient supplement. So what we did then in 2007 is we published a paper in patients with genetic mitochondrial disease. They have high lactate, reflective of their poor mitochondria, and they had elevated oxidative stress markers. So what we did is we used coenzyme Q10, which, as you know, is part of the mitochondria, shuttling from complex 1 and complex 2 to complex 3. It has antioxidant effects and also can be a couple there. We then put in alpha lipoic acid. So alpha lipoic acid is an antioxidant. We know it's in the mitochondria. It's part of branched chain keto acid dehydrogenase, PDH, et cetera. And the reason we did that is we wanted the two antioxidants to be a couple. So that's called a redox couple. They're in the same compartment of the cell. We also had vitamin E in that mixture as well. And vitamin E is there as a chain-breaking antioxidant in the membrane, because we have membranes around our mitochondria, around our cell in the sarcolemma. And then we added creatine monohydrate, which has some indirect antioxidant properties. It's present as an energy buffer in the cytosol, but it also shuttles into the mitochondria through the creatine phosphocreatine shuttle. So that was our 4-component complex. It was a randomized double-blind crossover study. And what we showed is when the patients were on that supplement, we call it multi-ingredient supplement of those 4 ingredients, we showed 2 markers of oxidative stress were lower and lactate was lower. So we got all sorts of criticism. People said, we don't know which one's working. And I said, that's the whole rationale behind this, is we don't want one. And so I used more of my own personal money, and I did a second clinical trial with extremely high doses of coenzyme Q10. So we used 600 milligrams twice a day. We had 30 patients, randomized double-blind crossover study, MR spectroscopy, VO2 max, $1 million study. And we showed absolutely no reduction in oxidative stress and no improvement in their lactate. So again, further supporting that to treat complex disorders, including aging, because aging is the mitochondrial disease we all have, I think that a multi-ingredient supplement approach is necessary, and it's critical that they're studied in humans in a proper clinical trial, so we really truly know that they're having the effect that we anticipate that they will have. Because sometimes you get the opposite effects that you really have to study.
Yeah, interesting. So, in the trial that you got criticism for, tell us a little bit about that study a little bit more. Who were the patients? How many patients were there? And were they healthy?
No. The first study in 2007 was MELAS. It was again mitochondrial.
Oh, they were the MELAS. Oh, got it.
Mitochondrial genetic disorders. There was some Leber's patients in there as well, but we had a very similar cohort in the CoQ10 study. So identical cohort of patients. Our multi-ingredient supplement was so powerful that even though the sample size was smaller in the multi-ingredient supplement, we had statistical significance in 3 metrics, whereas with the much larger study with 30 in a randomized crossover study, same group, mostly MELAS, but also things called MERF and Leber's, we showed nothing with just high-dose CoQ10.
Now, is this a commercially available cocktail?
Yeah, so the cocktail, what we did is, we said, look, we really should start targeting some of the more common disorders if we wanted to start a company and, get this to people, who might need this. So the first thing we thought about was obesity, because we know that there's secondary mitochondrial dysfunction, there's increased oxidative stress. All of this
Yeah.
theme keeps resonating,
Yeah.
in common disorders as it does in genetic disorders, as it does in aging. So we said, well, let's try. So we did some preclinical work where we essentially used a mitochondrial cocktail, which is, as we described. We also added beetroot extract. We then reviewed the literature and looked at 5 ingredients that are known to reduce body fat in humans, which was conjugated linoleic acid, green tea extract, green coffee bean extract, black tea extract, and forskolin, which is a mint extract. So we did preclinical work and showed that the mitochondrial stuff wasn't as good as we thought. Didn't do very much. So then we looked at the weight loss, those 5 ingredients, and it was very powerful. Now, we always throw exercise in, so that was our gold standard. So what we found, however, is that we then combined the 5 that I mentioned, the mitochondrial stuff, with those 5 weight loss, and we got this dramatic benefit. So it really synergized it. So we then started to pull some of them out. So we pulled out conjugated linoleic acid for complex reasons, pulled out creatine, pulled out black tea extract, and we ended up with 7 ingredients, which again, we didn't expect. The 7 were better than the 10. The 7 also increased something called UCP1, which is the jewel in the crown of everyone trying to treat obesity, because that's an uncoupling protein in the mitochondria, which sort of disassociates the food that you're eating with the production of energy, and you burn it as heat. So Bruce Spiegelman, who's a brilliant individual who discovered PGC1 alpha, started a company called Ember Therapeutics, again, ember burning, to try and upregulate this UCP1 and cause browning. Didn't go anywhere because, again, they used a single molecule approach, which is expensive and time consuming. But we found that with that combination of those 7 ingredients. Now, I was not comfortable, and I would not ever condone a supplement that could blunt the benefits of exercise, because I really think we all should strive for that as our gold standard. And we were pleasantly surprised that it actually doubled the benefits of the exercise. So that gave me confidence to move forward with a human clinical trial, which we did, where we used, and I don't know if you can see behind me if this is the video recorded, but
Yep. Yep.
Yeah, so we came up with what we call Trim 7. That's what Health Canada made us label it. We tried to use metabolic enhancer, which is what it did, but the 7 was the 7 ingredients. And we did a randomized trial in 60 overweight and obese men and women. And what was interesting is that, again, completely double-blind study, third-party GCP study. And what we found was a reduction in body fat, but a preservation of muscle.
So just to be clear, though, from the mitochondrial side itself, I mean, obviously you've got some other things in there as well, but from the mitochondrial side, you're really talking about CoQ10, creatine, and alpha lipoic acid.
Well, there's no creatine here. We pulled that out, actually, and we actually got the enhanced effect. So the UCP1 went up. So this does not contain creatine. It was just the alpha lipoic acid, CoQ10, vitamin E, and then it had those other components, the green tea, green coffee bean, and the forskolin. So that ended up being the most effective for that specific indication. That was to increase brown adipose tissue. I should say mitochondria in the white adipose tissue. They call it beiging or browning of adipose tissue. And we have other studies that we've done where we've taken that, we call it the mitochondrial core. That's the CoQ10, lipoic acid, and vitamin E. And we've added other components to enhance skin cells and older adult fibroblasts. We have worked with another group where we did some work for them to show that we could protect against radiation damage. So it depends on the indication. We think that core is necessary. And then depending on the indication, we tried various other combinations strategically and based on good biology to see if we can get a synergistic effect in the indication that we're looking for.
Yeah.
And in this case, it was lower fatty liver markers by almost 24%, as well as a reduction in body fat without muscle loss, which is unique, as you know, with Ozempic. Up to 40% is lean body mass loss when you lose weight through that mechanism.
Yeah. Just in terms of other commercially available type stuff that people are taking, just curious on your comments on there. One of them is, for example, MitoQ. I don't know if you know that one.
Oh, yeah, of course. Yeah. So MitoQ is just one of many ways in which people have tried to, twist the molecule, the coenzyme Q10 molecule, to either make it more bioavailable and, more targeted to the mitochondria. A lot of stuff has good evidence in vitro and even in preclinical models. But again, MitoQ is very similar to our high-dose CoQ10. So the type of CoQ10 we used was called Qzor, very bioavailable. And I think a lot of the things like itabenone, MitoQ, all came about when some of the CoQ10 molecules that were first being produced weren't very well absorbed. But we've largely overcome that. A lot of companies have very well absorbed CoQ10. So again, with our complete failure with massive doses of CoQ10, show me in a proper human randomized trial that it's actually doing what you say it's doing. And I just haven't seen that for that. And there's a few other ones. Itabenone is another synthetic molecule analog of coenzyme Q10, extremely expensive. Many of our patients are taking it for Leber hereditary optic neuropathy, even though the seminal study missed its primary endpoint. People are still pushing it for hundreds of thousands of dollars a year. And again, it's a single antioxidant. You don't get the redox couple. No. You're not protecting all the other parts of the cell. Yeah. And we have a paper coming out where we show that our combination approach with the MitoCore that we talked about was as good and in many cases superior to this very expensive itabenone in patients with Leber optic neuropathy.
There's a couple other ones just to run by you real quick. N-acetylcysteine and urolithin A are commonly cited, I guess, in sort of longevity spaces supplements. So
yeah, I mean, NAC predominantly, it's a cytosolic antioxidant, so you're not targeting the mitochondria, so you're not at the source where the ROS are being generated. There's absolutely no benefit whatsoever from a mitochondrial perspective. I don't know if anyone's ever tried taking it. Sure doesn't smell very good. We use it in the lab, and it's pretty gross stuff. And ultimately, for people to take things in the long run, it's got to be convenient. And if it's a powder or whatever, it has to taste good for the long run. Urolithin A, there's tons and tons of hype about this. And, what they have said is that you're purifying the mitochondria. So that whole concept I mentioned of mitochondrial autophagy or mitophagy, that's what they're purporting it is doing. If you look at the human study, though, I mean, really the seminal one where they're trying to purport that this helps people, what you can see is that they had 30 people in each group. So they had a placebo, they had a low dose and a high dose. And then at the end, where all of the hype comes about improving mitochondria and working by improving mitophagy, there were 6 patients per group. So what happened from 30 to 6 makes me a little suspicious.
Yeah.
Number 2, where they showed the dose where they showed increased mitochondria was not the dose where they showed increased mitophagy. With, again, I think in there it was only 5 subjects. So that's a little bit suspicious. So, from that perspective, it leads me, ho-hum. And what are you targeting with this? I mean, there's maybe a little bit of an increase in strength, but even when you look at that, like they had 2 metrics that were supposed to be measuring different things, and their dots for the patients were absolutely identical. So, raises a few questions about this seminal paper. And yet, in spite of it, people are, companies are buying this stuff and spending hundreds of millions of dollars on this, but the science is just not that strong, in my estimation.
Yeah.
Others may disagree, but you can take a look at that paper.
So, let's go back to the big driver, as you mentioned, is exercise for improvement. So what exercise regimen maximizes mitochondrial biogenesis ultimately most efficiently, with the least oxidative harm? I know you're an endurance athlete, but you hear a lot about high intensity stuff now, particularly as it relates to VO2 max, increasing VO2 max and that kind of thing. What's your take on that from a mitochondrial perspective?
Yeah. So a couple of things, it's pretty clear that the standard is moderate intensity, continuous exercise.
Like level 2 exercise, level 2 kind of stuff? Or?
Yeah, zone 2, a type of endurance exercise. That's what most of us had been doing for 30, 40 years. And even before me, the earlier exercise physiologists used that. And that's where, all of the work really showed the mitochondrial biogenesis. And there's no question that works. And then people questioned and said, well, I wonder if we do high intensity work. And you probably are familiar with Marty Gabala. He's really
had him on. Yeah, we had him on.
Marty's a good friend and colleague. I did most of the muscle biopsies for their study, and a lot of the mitochondrial work was done in my laboratory. So there's no doubt that it does. My only concern, and this is nothing against Marty or anything, but as somebody who's exercised his whole life, I find doing intervals challenging. So I only do them right in this before my season. And I don't, the rest of the year, I just love being out in nature and going for a run.
Yeah.
So I just wonder about the long-term sustainability. Number two, although they say it's really safe for older adults, it's pretty intense. Even me, now that I'm over the age of 60, I just find it a little bit tough to do high intensity stuff. And I just threw my back out the other day doing some high intensity stuff. So I guess that's the only concern I would have from a practicality. But on average, we compared moderate intensity continuous exercise to different forms of HIIT and sit, and there's all different ways you can skin that cat, and you get very similar increases. At the end of the day, I still say that all of our really strong epidemiological data on, lower cardiovascular risk, 4-year lifespan extension really comes from historical data where people were doing mostly moderate intensity continuous exercise, going for a walk, going for, canoeing, hiking, biking, cross-country skiing. That's really where we have most of our evidence. Now, the final point to that, I will say that with weight training, we were the first to show in older adults that there's almost a spillover. In when you're younger, it's very specific. You do weights, you get big muscle. You do endurance, you get mitochondria. But in the older adults, especially if they're sedentary, just doing pure weight training, we saw a huge increase in mitochondria. So I think as you get older, the specificity is not quite the same. I would also say, and we've done tons of work on this, but I hate to admit it, as I used to be, not world class, but raced internationally in a number of endurance sports. And as I got older, oh my goodness, I watched my muscles still get skinny, and I got weaker, and it was hard to pick things up. So really, as we age, we need to do both. We need to do both strength to keep our strength up, and we need to do endurance to keep our mitochondrial capacity and our metabolic health up.
Talk about the concept of an exokine.
Yeah, so exokine is...
or exokine, okay.
Yeah, so an
exokine.
Yeah, so I know how to pronounce it because I invented it. We were in a lab meeting, and it was Bente Peterson and Mark Fabreo. So I know Mark, we biked together. He's a great researcher, and Bente Peterson was the first person to come up with the concept of a myokine. And that was interleukin-6 produced in muscle, feeds forward to the liver, increases glucose rate of appearance. So they really came up with that concept. And I thought, wow, this is really smart, really great work. And so we were at a lab meeting, and it was, I think, about 17 years ago. And we were talking about their work, and, with my grad students, we were going over it. And, the concept of hepatokine and adipokines was coming out. And I said, wait a sec, all of these things, we don't know where they're coming from. Why don't we just call it an exokine? Anything that goes up in the blood in response to exercise, be it a protein, small molecule, or a microRNA, let's just call those things exokines. So years later, everyone tried to steal the term and thought that they came up with it. But, we coined it. We actually called our company Exokine. And, the first part of our company was to identify what these molecules are and see what they did. And interleukin-15 was the first one that we discovered, which mediates the muscle feeding up to the skin to increase mitochondrial biogenesis in skin.
What are you seeing with that on a practical basis? Are you using exokines for anything right now, like in your clinic?
Yeah. So essentially, that was the whole premise of our company. And we ended up going bankrupt as a consequence. So what we did is we first identified interleukin-15, and we have the Canadian US patent. A couple of things happened. Number one, we got all jazzed up about exosomes as being the carrier. We and others, there's various places where you can look and see what's in an exosome. IL-15 is clearly there in exocardia and such like that. So we were thinking maybe all of these exokines are being transported with exosomes, and it's really the exosomes that are the important thing. And then we knew that nucleic acids could be in exosomes, and as a neurogenetic specialist, I thought this is great. Maybe we can deliver mRNA and other things to tissues with exosomes. So we spent a lot of time, a lot of money going down that pathway. And a couple of things are pretty clear. Number one, exosomes don't do what people think that they're doing to a large extent. And although fragments of mitochondrial DNA and sometimes even full-length mitochondrial DNA are in them, it doesn't really exchange between tissues. And what made our company go bankrupt was one of my patients who had MELAS syndrome, and he had a bone marrow transplant. So in his bone marrow, he had a new, what we call mitochondrial haplogroup. So we all have slightly different mitochondrial DNA, which reflects when we shared a common mom. So we could now trace the exosomes from his bone marrow into his muscle, into his connective tissue, and into his endothelial tissue. And what we found is after 2 years, not one of the exosomes, which are just floating around like crazy in his blood, made it into his muscle. So although he was bathed 24/7 for 2 years with exosomes that we isolated, clearly showed that the mitochondrial DNA from the donor was there, none of that went into the tissues. So all these people that think that you can deliver DNA and RNA with exosomes, it just doesn't work. So yes, we kind of abandoned that. Also, taking a small molecule like IL-15, and then injecting it into a human to mimic the pulses of exercise becomes a very expensive pharma development. And, to raise $25-30 million, like Bruce Spielman did for Ember Therapeutics as a small molecule, one fit fails and you're out $30 million. So we kind of abandoned that. Yeah.
Yeah. What are your thoughts on, some of the other novel therapeutics for mitochondrial enhancements like SS30, or some of these peptides like SS31? I guess there's MOTS-C as well. What's your take on those?
Yeah, I mean, the challenge with a lot of these things is, number one, it's a single molecule. And the challenge we have there is, one, we have to figure out what's the concentration that fits with Mother Nature. Maybe I'm giving away too much here, but for IL-15, the reason why nobody thought about it is that you do a transgenic animal overexpressing it, and it's, 10 orders of magnitude more than what's in physiology. So what was unique about our study is we then went back to Mother Nature and said, what does IL-15 do when we exercise, knowing that exercise is your biological default? So we figured out what the pulses were with exercise so that we weren't giving a chronic inflammatory response. We pulsatility induce the IL-15. So we figured out how much to inject in the mouse to pulsatile, very similar to what they got with exercise. And that's when we saw the benefit. So I think, and this is again a concept, if we ever raise a bunch of money, I'd love to, which we've already done. We've profiled all the exokines that go up in response to exercise, figure out how much you would have to administer, very much like an insulin pen, and give, small pulses of a whole number of them together. The problem is that our traditional pharma model doesn't like that. They want to identify one target, one molecule, find out the PK, go through your standard phase 1, phase 2, phase 3. And that's why they're all going to fail. Like apelin, you probably just are aware, is an exokine that just failed in large-scale clinical trial. I don't think single molecules are going to work like our multi-ingredient supplement approach. I think the only way that these are going to work is to mimic Mother Nature and have 5, 10, or 15, but it doesn't fit into the pharma model. And so investors will not invest in this stuff, even though it's the only thing that's going to work in my estimation.
What do you think about the concept of infusions of mitochondria? I know I vaguely remember something about, platelet-derived mitochondria, maybe taking your own cells and culturing them and, basically getting these mitochondria and infusing them back in, or any other sort of mechanism by which you're basically infusing mitochondria.
Yeah, so we know for sure that the mitochondrial DNA doesn't work. And we know that from my patient. I know it's only one patient, but we actually had a second bone marrow transplant patient and showed no transfer of the mitochondrial DNA. Now, that doesn't mean that the mitochondria themselves are not the issue. Now, there's a couple of things to think about. I mean, clearly people have shown with some, cardiac damage models, that you can, for example, take a muscle biopsy, culture it, extract the mitochondria, infuse them back in, and they can help to repair the damage in the myocardium. I mean, that is really good, strong evidence for that being the case. So I think in certain circumstances, if the tissue is damaged, it seems to be able to incorporate intact mitochondria. I think as we move forward, we need to have very good GMP facility to manufacture the mitochondria so that we don't break them open, because if the mitochondrial DNA is released, it activates what's called the inflammasome. And so it could be a very toxic infusion. So, some of these people who are doing it in third world countries and they're going down for exosome infusions and all this kind of stuff, it's probably snake oil because they're not getting allergic reactions. And certainly exosomes, can activate the immune system, which is one thing that we discovered. But the same thing I'd be worried about is people, taking off and say, I'm just going to infuse a whole bunch of mitochondria. If they break open and you release that mitochondrial DNA, it's an issue. The other issue too is that although our mitochondrial DNA is very similar in terms of the genetic sequence, there's a thing called a haplogroup. So if I sequenced your mitochondrial DNA and mine, there'd be base pair differences, which reflect when we shared a common mom. And those also, to some extent, are almost a form fruit of self and non-self activation of the immune system. So, what haplogroups are compatible? What I mean by that is that if you take another person's skin cells, grow them, and they have a different haplogroup than you, could there be haplogroup incompatibility and other issues? So it's a really complicated area, but it definitely is exciting, and there's people definitely working in that area. And I think it's definitely has a role for therapeutics.
Yeah, and one of the other, so one of the things that comes to mind to me, I guess, is like when people are hearing all this and talking about, trying to improve mitochondrial function and all that.
Yes.
How do you, as an individual, how can you measure how, if you're actually making improvements in your mitochondrial health? Like what lab tests can you take, or are there other ways to kind of get a sense for your mitochondrial health?
Yeah, I mean, there's innumerable ways that we can do that in the laboratory. From doing muscle biopsies, punch biopsies, looking
Right.
at different
But just like,
markers.
people
For
listening to the show,
yeah. I mean, I think the best thing, tried, tested, and proven, we know it's associated with improved health span and lifespan, is your VO2 max. And the watches are getting better and better at being able to give you a reasonable estimate for your VO2 max. And I would say for, anybody who does not have access to, high level biochemistry and molecular outcomes, VO2 max really is such an important metric. We know for sure that really low VO2 max predicts institutionalization. Higher your VO2 max, the better your health span and lifespan. So get a good quality watch. And, ideally, if you could, I think for most people, it'd be really nice to take your watch, get a good body composition measurement as well, and look at body comp and look at your VO2 max. So you do a VO2 max test with your watch so that, your watch then could be calibrated to a very accurate VO2 max test. And then also get a body composition done with a very accurate test called the DEXA scan. And then at home have what's called a bioelectric impedance. And for ultimate health span, I think, just follow your VO2 max on your watch.
Yeah.
And use what's called a bioelectric impedance, where you have both your hands and your feet, not just the ones with just your feet that you see in most gyms, but there's ones where it's your hands and your feet. Gives you a better representation of your body composition. And those really, for most people, are going to tell you about healthy body composition changes. Because if people are a slave to the scale and all they care about is weight, you can be misled. So you can lose a lot of weight, but you may be losing lean body mass. Like Ozempic, like even fasting, you lose up to 25% of the weight loss is lean body mass. So a good way to assess fat versus lean mass, and a good way to assess VO2 max, I think, is the simplest way for people to really follow their health span.
So we talked about exercise, and the last thing I want to just mention, and it is something. So I interviewed Dr. Robert Lustig one time. I'm sure you're aware of him, endocrinologist, UCSF guy, and he's pretty vocal about diet, and he talks about metabolic dysfunction stemming from the food we eat. Usually, when I think of metabolic dysfunction, usually I'm from my clinical training, I usually think of insulin resistance and that kind of thing. But Lustig actually says this metabolic dysfunction, at the level of the mitochondria, can be triggered by various processed foods. What are your thoughts on that?
So I think there's a couple of points to make there. I think that diet is absolutely critical. And I think I subscribe to The Economist magazine. I think it's very well written. I espouse many of their philosophical constructs, but I vehemently disagree with the front page where they're talking about Ozempic. And that was eat, inject, repeat, curing obesity worldwide. That was the title page that everybody saw in The Economist. And what they had was a hot dog. So essentially what that tells people is don't worry about exercise. Don't worry about what you eat. Eat whatever crap you want. Take this Ozempic and it's going to help you. That is the worst message, I think, from a public health perspective. But again, it's what the drug companies want. They
Right.
don't want exercise because it's going to take away market share. They don't want good diet because it takes away market share. So they love that, message to go out. But I think it's completely misleading. I think we need to focus on improvement in muscle mass, strength, endurance, VO2 max, and, as you pointed out, nutrition. And nutrition, there's some simple things people can do. Eat, locally, eat as close to nature as you possibly can. Have a lot of color in your diet from fruits and vegetables. All of those things I think are important. Protein is really important. So too many of us, as we get older, you've heard of the tea and toaster, where people are essentially having tea and marmalade, toast and marmalade, and then a bagel with, whatever, a bit of butter on it or God knows what. And the quality of our food, I think, is really deteriorating. The problem there is crappy food is cheap. Good quality food is generally more expensive. So it's a huge effort for people to eat well and to eat a variety of foods. But if, you have the wherewithal to do it, and I think, that's where a challenge we're going to have with these tariffs and other things is, it's going to make imported foods more expensive. And certainly for us in Canada, in the winter, if we don't import our fruits and vegetables and these sort of things, people are relatively going to be in a state of nutrient deficiency. And I think that, the biodiversity of our microbiome critical from having a variety of different foods. And this has huge metabolic benefits. And if we eat processed foods, lots of simple sugars, we simplify our microbiome diversity, which leads to all sorts of metabolic consequences. So in many ways, I completely agree with what this individual is saying.
Great. So Mark, tell us a little bit about where, people can learn more about your work, more about this topic that, and continue to learn.
Yeah, well, certainly I think for most people, there's so much garbage out there. There's so many claims and people are hand waving. I would encourage everyone, of course, to go to PubMed and, type in the person's name. If you hear about a study, check for it in PubMed. If something is not in PubMed, then I really wouldn't pay it much attention. So what we mean by that is in PubMed, you're looking for a peer-reviewed article. So your peers have ripped it apart and criticized it. And so you have to defend what you're doing. And that's really the main place to take a look. So,
How about in terms of your work?
that's what I'm saying. Just go to PubMed, type in
Oh.
the
Okay. Got it.
Yeah, I
And
think that's the
great.
best way for anyone to,
Yeah.
to take a look whether something is credible or not.
Fantastic. Thank you so much for joining us.
Oh, my pleasure.
Thanks for listening. A quick reminder that while I am in fact a surgeon, nothing I say should be construed as medical advice. Now make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.